Hello everyone, my project was over canine distemper virus. Today we will be mainly focusing on the cross-species infection, along with the vaccination against the virus and the current antiviral research that has been happening. I hope you'll find this very interesting. Okay, to start off, let's talk about why it's important to know things about canine distemper virus. This infection covers a wide variety of animals, such as red pandas, um, canids, felines, except for domestic cats, hyenas, raccoons, seals, bears, etc. You can see a full table over here on the right of the screen. The mortality rate is typically 80% in puppies and 50% in adult dogs. However, once the neurological issues set in, the mortality rate skyrockets significantly to almost a 100% chance of death at that point. It is highly contagious, so there are a lot of conservation concerns, especially for those endangered animals such as pandas and leopards and tigers in research facilities and those breeding programs. There is the issue of cross-contamination with outside animals. There's currently no antiviral treatment. All we have is symptomatic treatment and preventative measures for secondary infections, which can cause an even worse kind of symptomology and worse prognosis for the animal. And then the other important issue is that it has the potential to spread to humans, as it has been reported in our closest non-human relatives, including rhesus macaques and baboons. So that is a very real possibility. So typically there are two phases in the infection progression. The first phase is called, or typically referred to as the acute phase, where within 24 hours after infection, the virus starts replicating in the lymph nodes and macrophages, which are our immune cells, where it leads to immunosuppression, severe immunosuppression. And from there, it travels through the lymphoid system into the respiratory tract. And roughly 6 days post-infection, the GI tract is infected in the epithelial cells, along with the spleen and some specific kidney cells called Kupffer cells. That's when it starts replicating within there and will continue to spread even further. Roughly a week after infection, the viral shedding begins. So this is when the animal is contagious. It can then spread the virus through sharing of food, sharing of water, excrement, pee, if they come into contact with that. There's also the issue of potentially if it consumes or if it is consumed by another animal once it's contagious, it can cause that to spread that way. And roughly 2 to 4 weeks after infection is when the neurological issues set in, and the death rate tends to be fairly high. So we usually see the mortality happen within 2 to 4 weeks after infection. So some general symptoms that can happen is that biphasic fever, which is just the fever that has the onset on the two phases, both the acute and the late phase. It usually happens during viral replication in the lymphatic system, and then while it's replicating in the central nervous system. They also have issues of eye irritation, so conjunctivitis, dehydration, depression, trouble breathing, eating issues, gastrointestinal issues such as vomiting, diarrhea, anorexia. Like I said in the last slide, severe immunosuppression. There is the trademark hyperkeratosis, which is the hardening of foot pads and the nose due to an increase in keratinocytes, and it usually shows up in kind of those little fibrils on their paw pads there. There also can be lesions or pustules. They typically happen around the abdomen and thighs in the acute version or in the acute phase. and the animal will get severely lethargic. There'll be a lot of discharge from the eyes and the nose. There is general posture issues, so they'll typically just end up laying on the ground, and eventually their kidneys will start to fail as well. So during the late phase of an infection, we typically see these neurological symptoms such as chewing gum fits, which is the repeating clacking motion or chewing motion of the jaw that doesn't necessarily result in lack of consciousness. There's a circling behavior. They have constant crying, incontinence issues, muscle spasms, seizures. They can even have paralysis at some point. A lot of this is onset of the impending death of this animal. So once the infected animal gets to this point, there really isn't much that we are able to do. So I want to go over the neurological aspect really quickly. There are two known ways of the virus getting into the central nervous system. So after entering the airways, the virus infects those lymphocytes and the immune cells and then spreads throughout the rest of the body by attaching to specific receptors, including Nectin-4 and SLAM receptors. And then there are two options that can go from there. There's the retrograde infection or heterogeneous dissemination, where the virus circulates its way through the cerebrospinal fluid and up into the blood-brain barrier, where it then destroys it, allowing it access to the central nervous system, where it then replicates within the neurons and astrocytes. Or there's the other option, which is the anterograde infection, where the virus will travel along that olfactory nerve, typically in the direction of signaling up into the brain, and it will infect the neurons from there. Typically it has a non-cytolytic spread, so that means that while it spreads, those cells tend to not die off. It has a very selective spreading that once it's in the brain, it typically only occurs at the tip of the astrocytes. And then the infection leads to disruption in myelin production, and that then leads to neuronal death, which can cause the symptoms that we saw on the previous slide. I just want to touch on some key aspects of the cross-species portion. It is a highly contagious disease. It has the potential to become zoonotic, however, because it hasn't crossed over to humans, we can't call it that yet. There are genetic mutations that have allowed it to infect non-canid species, one of which is thought to be the Y549H substitution, which we'll talk about in the next slide. Because of its high communicability, there are heightened conservation concerns, like what I said before, and we call the infected pool of animals in the surrounding area reservoir species. These species can differ based on the target of the certain study, such as wildlife versus domestic animals. Depending on which species you're looking at, it can vary there. These species are key to the spread of the virus because the virus doesn't survive long outside of a host, and the infection follows a very similar progression pattern across species in general. So it may vary a little bit in the progression rate in general, but overall it's the same. So to go a little bit more in depth about that substitution that I talked about in the previous slide, there's not a lot known about it. We do think that it has some aspect for the cross-species infection. However, all that we really know is that there is a residue change on the H protein at the 549 position, where the tyrosine is replaced with the histidine. The function of the substitution isn't really known in general, and it's usually seen when the outbreak animal is non-canid. However, this is not a strict requisite for this substitution needed for those non-canid species. Hypothetically, if we learn more about this substitution, we may have a better understanding of the genomic evolution of this virus, the host selectivity mechanisms of the virus, and how it interacts with the host's immune system. So current preventative measures are vaccinations. We typically vaccinate domestic animals such as dogs and ferrets, and these vaccinations of those domestic animals helps limit the spread to those reservoir species. One would think that, well, why don't we just vaccinate those wildlife to help reduce it even more? However, there are shortcomings to this. One is due to the type of vaccine that we have. It's a modified live virus, so it needs to be very species specific, and doing that is very costly, not only in production, but just making it in bulk enough for those. There's also a general lack of interest in the United States to make a non-domestic vaccine, and it's just our current methods are just impractical. The catch and release is costly, and the tracking of those is very difficult. And the captivity efforts that we have done, oftentimes the animal is not eligible for release after that series is complete due to that prolonged time in captivity.
So as we're wrapping up here, I just want to give a general overview of our antiviral. There is no clinically available antiviral treatment. We only have the symptomatic treatment and prevention of secondary infections. However, there are currently experimental compounds such as ribavirin, interferons, and a couple of other things that have previously been shown to be effective in diseases such as measles and other related viruses. There have also been studies done looking into the effects of combining these, such as ribavirin and interferons, to potentially make a safer, more effective treatment. However, none of these have been officially approved for use in a clinical setting. The other portion of this antiviral thing is that they all impede on the viral replication in some way, whether that be the general replication process or the transport of those virons that are resulting from that. There is a major complex here in the makeup of canine distemper that we call the RDRP complex, which is known as the RNA-dependent RNA polymerase complex. It is a very key aspect in the viral genetic replication. However, we don't know anything other than that this complex exists, really. So theoretically, if we learn more about the mechanisms and the actual makeup of the RDRP complex, it can help expand our understanding of the antiviral mechanism itself, so how it interacts with that virus and helps limit viral replication. And it could potentially provide a new target for more effective antivirals, as stopping that replication before it even happens would be more effective than allowing it to replicate and then preventing it from transport throughout the body. The other issue for this is that each RDRP complex is unique to the virus. So we have had similar antivirals that target the RDRP complex, such as ones used in influenza. However, this cannot be applicable to CDV itself because of those differences.
So since we're wrapping up here, these are all the sources that I used for this paper. I have a total of 44 sources in total, but all of those are listed in the thesis itself. Obviously, there isn't enough room for them on this slide, so I just picked the main ones that I used within this presentation. And thank you so much for coming and listening, and I hope that this was informative and you found it as interesting as I do.